Hello, everybody. It's 12 in Warsaw, and we are starting our UNIgreen JRC4 seminar. And before we proceed with the presentation, I wanted to briefly introduce you to what we are joining today. So we are here under UNIgreen, the Green European University, which is a project co-funded by European Union that gathers 8 universities around Europe. One of them is Polytechnic University of Coimbra, which will be having a guest lecture today as a seminar, presenting the seminar by Manuela Abelho. Then we have other universities from Spain, Italy, Bulgaria, Belgium, France, Iceland, and Poland. And together we form UNIgreen, this alliance of universities. And inside UNIgreen, we have joint research centers, one of which is organizing this seminar, the Joint Research Center number 4, which is focused on biodiversity, forestry, and agroecology. And today's meeting is planned to be in a system that we will have a half-hour presentation followed by a discussion. And this discussion will take place by Q&A that you have. You can use on the top of your Teams window. So there's this Q&A button using which you can ask questions to our presenter. The questions later at the end will be read out, and we'll try. I think Manuela will try to answer them for sure. And what else? The whole meeting is being recorded, and the recording will be later shared for the wider audience for the purpose of teaching and also to spread the scientific knowledge. And I think that that's all from my side, because I don't want to make it too long. I'd rather go for the presentation itself. So the presentation will be about water ecosystems, and I'm not going to tell you any more details. I'll let Manuela say more, and I'll give floor to her. Manuela?
Hello, good morning, everybody. Well, it is about freshwater ecosystems, more specifically. And during my presentation, I hope not to take more than 30 minutes. I will try to convince you that these small rivers or streams are really, really cool to study. Let's see if you agree with me. So I have a title that maybe for the people that do not work in these ecosystems may sound a little bit strange, but I will try to explain as we go on. So what I do the most is studying leaf litter decomposition. So I study leaf litter decomposition, and the most interesting thing is that I've been studying this for, well, many years, since 1992, in fact. It's almost terrible to think about how many years. But why is this so interesting? Why are these ecosystems so interesting? This is not moving. Okay. So these are images from the streams that I study, the type of streams. And as you can see, they are usually shaded. They have riparian trees. Riparian trees lose their leaves. And so I've been studying the ecosystem surrounding these leaves in the streams, which in fact are really important for the system. It's very slow moving. Okay, so why are these streams so interesting, or these small rivers? So as you all probably know, most ecosystems rely on producers as the basis of the food webs. And this goes for almost all ecosystems except for these ones. And so they are really, really interesting. Even if you think about the deep of the oceans, they still rely on producers for the basis of the food webs. But in these streams, there are almost no producers. So inside the water, you can see the producers outside, but inside the water, there are no producers. And this is because there's limited sunlight entering the water. And because of that, even if there are a few primary producers growing, they are not enough to fuel the food webs. So instead of the food being produced inside the ecosystem, these ecosystems rely on the organic matter produced outside of the system. So the leaves that shade the water also produce the fuel for the aquatic food webs. These leaves fall into the water, are decomposed, and then introduced into the aquatic food webs, ending, for instance, in fish. So how does this happen? So we have the riparian leaf litter, and in autumn, at least in our climate, in autumn, the leaf litter falls from the trees, and some of this leaf litter falls into the streams also. And we can see in this graph that the highest litter fall is around autumn, and the rest of the year has very few amount of leaf litter coming into the stream. And this leaf litter, of course, is composed from everything that falls from the trees. But as you can see in this dashed line, it's mostly leaves and not other things. We have total, we have woody debris and also reproductive parts like fruits and flowers, but it's mostly leaves. So these leaves are the basis of the food webs. The food webs use this litter as food. And this is consumed by some invertebrates that are called detritivores because they eat the detritus. So later on, the leaf litter is colonized by decomposers, and these decomposers in running waters, because it might be different in Atlantic waters. In running waters, these decomposers are mainly a type of aquatic fungi that are called the aquatic iphomacetes, and they colonize the leaf litter very fast. You see, a few days after falling in the water, it's already colonized. They colonize, they grow, and then they disappear really fast also, meaning that in later stages of decomposition, they are replaced by bacteria. So you can see here we have an indicator of fungi, and fungi increase and decrease. And here we have ATP, which indicates life. And so we have the fungi in the beginning, but later on, there's still some life there, microbial life, but they are not fungi anymore, they are bacteria. And with this, they decompose the leaf litter that it's then eaten by other organisms. And these organisms, the detritivores, are mostly insects. Many people don't know, but many insects have aquatic stages during the larvae stage. And so they are mostly insects. They are taxonomically diverse. I have here a small sample, but they are really very diverse. And most of them eat the leaves, which is not surprising because there's not much else to eat. So the leaf litter is then decomposed by the microorganisms, and the velocity of decomposition depends on several things. One of the things, of course, it's the litter itself. It has been proved that some species disappear much faster, they are decomposed much faster, while others decompose slower. It also may depend on if the leaf litter is offered single or in mixture, and I have here some single species and some other in mixture. And you can see the rates of decomposition vary among the species, but also among the mixtures. So mixing the litter may have different effects from the individual species. And of course, some other things that are also important are the stream characteristics, the current velocity, if it's flowing faster or slower, the physical-chemical properties of the water, the temperature of the water, which also depends on the geographical area. So this all influences the rate at which the litter disappears in the streams. So, but the title says riparian-stream meta-ecosystem. So, as I tried to explain, these small rivers depend on the terrestrial vegetation, and in fact, they are a continuous. So it's a meta-ecosystem, not two separate ecosystems, the terrestrial and also the aquatic, but there's a place, the riparian area, which is a connection between the two. So how does this happen? So we have the leaf litter that falls directly into the stream. When the trees are above the stream, of course, they may come directly into the stream. And when this happens, the leaves are freshly excised and there is only aquatic decomposition. However, it has been proved also that some of the litter inputs do not come vertically. They first fall on the riparian area, and then they are, when we have, for instance, a storm or precipitation, they are, after being at the terrestrial ecosystem, they are carried out on into the stream. So we call these the lateral inputs. However, most of the studies made with leaf litter decomposition assess only the vertical litter fall and what happens to decomposition of this litter fall. So, but we know that some litter come from the lateral inputs, and this may mean that there already was some decomposition while they were in the terrestrial ecosystem, meaning a mixture of different ecosystems influencing what happens to leaf litter decomposition. Usually, people do not study these lateral inputs or the decomposition of these lateral inputs because there's an historical separation between the terrestrial ecologists and the aquatic ecologists. And this, although it's very old, it still continues. We either study aquatic ecosystems or terrestrial ecosystems, and mostly people do not join both ecosystems. So I'm going to show you, if this works, I'm going to show you an experiment where I tried to join both ecosystems. So I carried out an experimental design where I had a mixture of several stages mimicking what happens when the leaf litter falls directly into the water, so fully aquatic decomposition. Okay, fully aquatic decomposition. And then I have a mimic of what happens to the leaves while they flow, they are carried out into the water. And so I did this by having, in total, the experiment took 56 days. And so in the first case, there was no terrestrial exposure, only water. In the second case, there was 14 days of terrestrial exposure, followed by 42 days of aquatic exposure. And finally, in the last case, there was half-half, 25 days in the terrestrial ecosystem, followed by 28 days in the aquatic ecosystem. And what I'm going to show you is that probably what we've been doing most of our lives, people that study aquatic decomposition, do not represent fully what happens in water. So, showing the decomposition rate, I had 2 leaf species and a mixture of both leaf species. And here we can see that the leaf species decompose at different rates. We already knew this. Some species are preferred and some species are not. Here I have the decomposition rate. So the higher, the faster the leaves disappear. And you can see that alder is very fast decomposing and the others are slower. But most importantly, what I wanted to show is that what happens to leaf litter depends on the exposure. So fully aquatic exposure is much faster. And while when we have a period of terrestrial exposure, decomposition goes much slower. So there is a decrease in decomposition as we increase the time, the period of terrestrial exposure. This happens probably due to moisture limitation here compared to in stream, and also to reduce leaching of the compounds of the leaves. Here is much slower than here. And also it has been proved that terrestrial detritivores have much lower turnover rates than aquatic detritivores. And the faster aquatic decomposition rates, of course, are promoted by the water velocity. It's a big important factor in decomposition because also the leaf shredding invertebrates are much more efficient here than in the terrestrial ecosystem. And also there is the aquatic aphomacids that it looks like they are also more efficient than the terrestrial fungi. Regarding fungal colonization, we can see a clear tendency also that sporulation, which tell us the reproductive activity of the fungi, it was much higher inside the water than, in the fully aquatic exposure than in the exposures with terrestrial exposure. And also the taxa richness was much higher in water, totally in water. And of course, this is due because we studied the aquatic ipomycetes. So in all cases, they were always in water, but it happens that here they were always in water, while here they were first in the terrestrial ecosystem. So we can see that probably this totally aquatic exposure means that probably the fungi here invested more in reproduction than these ones. Also that probably this litter was already colonized by terrestrial fungi, and these terrestrial fungi were not able to sporulate in water. It happens that some species can sporulate, some terrestrial species can sporulate in water, while others don't. And also, it looks like that litter that was previously exposed in the terrestrial ecosystem may not be a suitable substrate for the aquatic ipomycetes. But this means that when we only study this picture, the fully aquatic exposure, we do not get what really happens in nature. So this experiment also included the measure of invertebrate colonization, and we have invertebrates in water, but also outside of the water, and so they were all counted. But you can see that there was a decrease in the abundance of the leaf-eating invertebrates, more abundant in water and less abundant in the previously exposed to terrestrial ecosystems. But when we look on their biomass, we can see that they are less when there's a previous terrestrial exposure, but they have higher biomass, meaning that they are less in amount, but they are bigger. And bigger, they might eat as much as many small ones. So, but the title that I chose also includes this sentence that the journey is as important as the destination, because what I'm trying to say is if we study only the litter that falls directly into the stream, we do not take into account this journey that the litter might have from the terrestrial ecosystem into the water. And so we can see that the differences are big between the exposures, meaning that most of the studies were based only in fully aquatic exposure, and they do not tell us everything about what happens to these leaves. Maybe I'm speaking too fast because I'm already approaching the end. Okay, so I have a second part, and this second part is I called a sequel because I still want to know what is the effect of a terrestrial exposure before the leaves enter the water. And I wondered if this exposure in terrestrial ecosystem before entering the water would change also some other things that we, the aquatic ecologists, usually study, which is the effect of mixtures. For those not familiar with the effect of mixtures, I will try to explain. The studies we carry out, because it's simpler, are usually carried out with a single leaf species, but we know that in nature, when leaves fall from the trees, they mix. And so when they arrive to the terrestrial or to the aquatic ecosystem, they arrive in a mixture. So many people have already studied these effects, the leaf mixing effects, that's how it's called, and it may happen two things. So these leaf mixtures may decompose at the same rate as as the average of their individual leaf species. And when that happens, we say that the effects are additive. So we can find the response of the mixture by averaging what happens to the individual species within the mixture. But in fact, in many cases, it has been proved that decomposition of the mixture is different from the average of the individual species that compose the mixture. And when this happens, we call these non-additive mixture effects that can be either synergistic, meaning that the mixture decomposed faster than the average of the individual species, or they can be antagonistic, meaning that the mixture decomposes slower than the average of their individual species. It's already well established this. What I really wanted to see was if this mixture effect would change in leaves that were only exposed in water and in leaves that were previously exposed to the terrestrial ecosystem. And to make it short, there are more results. I will try to explain what happened. So I have these graphs, and first I'm going to explain what these graphs are. They are a graphical expression of the leaf mixing effects that are easy to understand, because what we have is we have a line where there is no difference between the mixture and the average of the leaf species. And then we have, this is the average of all replicates. And then we have the lines, which are the 95% confidence intervals. And if the 95% confidence intervals do not cross the 0 line, then the effect is additive, meaning that the mixture decomposes the same as the average of the individual species. But when the 95% confidence intervals do not cross this line, then the mixture has non-additive effects, meaning that it might be decomposing faster than the average of the individual species or slower. And here we can see what happened in the 3 things that we measured in these experiments. So we measured mass loss, fungal colonization, and shredder colonization. And as you can see, there's a clear difference between the 3 exposure scenarios. So we have fully aquatic, partially terrestrial and mostly aquatic, and then half terrestrial and half aquatic. And we can see that the leaf mixing effects, in fact, change across this continuum from the fully aquatic to the more terrestrial exposure. So we can see that in the case of mass loss and fungal colonization, they are additive when we have any terrestrial exposure. So a short period or a long period resulted in additive effects, meaning that the result of the mixture was similar to the average of the 2 species. But in fully aquatic exposure, we can see that the mixture has a synergistic effect, meaning that the decomposition of the mixture was faster than the average of the 2 species. This happened for mass loss and happened also for fungal colonization, which was higher in the mixture than the average of the single species. Regarding shredder colonization, we have a different result, but also shows that the exposure, the type of exposure really influences the results. So in this case, we have that in fully aquatic and half terrestrial exposure, there's an additive effect, meaning that the number of animals colonizing the mixture was the same as the average of the 2 species. But in this exposure, which was only a little bit terrestrial and mostly aquatic, we have also a synergistic effect. So we can see that when we study only what happens to leaves that fall into the water, the fully aquatic exposure, we have a good picture of what happens when the leaves fall directly, but we do not know some of the things that are happening in nature, which is the leaves that fall first in the terrestrial ecosystem. They start decomposing and then they go slowly into the water and finish decomposition. And we can see that this journey across this continuum really alters what we know about, what we thought we knew about leaf litter decomposition in streams. And hence the title I chose, The Journey is as important as the destination, meaning that along the journey, many things happen that we, when we study only the full aquatic exposure, we do not capture all these differences that this study shows. And with this, I speak really fast. So I end and I will answer some questions you may have.
Great, thank you, Manuela. Thanks for this really interesting presentation. I think that we now can proceed to question and answers. So I would encourage everybody to ask questions using the Q&A panel that you have above on MS Teams. And while you'll be asking questions there, I have the privilege of asking the first one, giving you some time to think. So I think I would like to start with a methodological question. So these three different options, 100% aquatic, 25, 75, 25, and 50, 50. I understand that all those research were made in the field, right? I mean, in the
field.
Not in the lab, right?
In the field, all field experiments, yes.
What was the amount or the number of the replicates that you did? Because you mentioned that there were some replicates.
Yes. So for each, what we do in these experiments, I will explain first. So we prepare a lot of bags. We call it leaf bags, and these bags they have holes for the animals and the microbial organisms to penetrate. And so we measure first the amount of leaves in the beginning and then in the end to see what happened during the period. And in this case, we had 3 replicates for each species and also for the mixtures. And in the case of the terrestrial exposure, we had to have extra replicates because we sampled before and we had to assess what happened in the terrestrial period, and then we have to continue. So we have more replicates so that some would continue. They were, in fact, moved into the water, but of each condition, 3 replicates.
Okay, so then all of them were done in the same place, same area, right?
In the same place, but
similar conditions.
Yes. So first, the ones in fully in water, they were put inside the stream. I can explain if you want. And some were put outside the stream. And so the ones outside and the ones inside were collected periodically. And then after 14 days, some with the necessary amount of replicates were transported from the riparian area to the stream. And after 28 days, another were transported from the riparian area into the stream, but with the necessary amount of bags so that we would have the same 3 replicates each time we went to collect.
I would be afraid that I'm going to lose some of the bags. Did you lose any? I mean, you know.
The bags. Oh, in this experiment, no, but I often lose a lot of bags. In the terrestrial ecosystems, there are no problems unless there are some animals or people, usually people taking. But inside the water is terrible. Many of my experiments, because all of this happens mostly during autumn, and during autumn is where we have the highest precipitation, at least here in Portugal. And so sometimes the current velocity is so fast and it goes so up, the water goes up. And sometimes I end with the bags in the trees, on top of the trees and everywhere except where they should be. It's very difficult to work in streams in winter, I tell you.
Okay, that's really interesting. Okay.
Everybody is ashamed.
I have more questions, so while we be waiting for any to be asked, I don't see any. If anybody wants to ask question, you know, out loud, I can give you permission to speak, but maybe written ones. I don't see any yet. So while we'll be waiting, I wanted to ask you this question. So the decomposition rate was slower each time there was any terrestrial ecosystem involved.
But it's also very well established already, because as I tell you, we work separately. The terrestrial ones study the same things, but only in terrestrial ecosystems, and we study the same thing, but only in aquatic ecosystems. So we already knew that decomposition in the terrestrial ecosystem, it's much slower than decomposition in water. And this is mostly due to the effect also of current velocity when we speak in flowing water. But also the fauna, the microorganisms and the fauna are more efficient in water than in the terrestrial ecosystem.
Yeah,
so
but I wanted to ask you if, so for example, some microorganisms have colonized the leaves already on the terrestrial ecosystem, and then you move them to water. So do you think that
I think that some of them will die.
Yeah, any kind of fight between those ones?
Yes, also. Some of the terrestrial ones will not survive in water, some will. Even the ones that survive in water, many will not be able to reproduce, to sporulate. And I think there's an antagonism between the two communities, yes. Although I did not really measure, I cannot say for sure, but I think, yes, that the two communities may be antagonistic, yes.
Because I was wondering if this partial exposure to terrestrial ecosystem might, for example, slow down the decomposition rate in water for first period, let's say.
I also I don't show, but I also measured that, and it slows down really a lot.
So you were measuring along the time? I mean, did you measure?
And so I measured the decomposition rate while it was in water and after separately. And a period of terrestrial decomposition may slow down what happens in water. So overall, the leaves that were first in the terrestrial ecosystem, they will decompose slower.
That might be this antagonism on one hand.
Yes.
What would be the other ecological explanation?
There are several, in fact, because in the beginning, decomposition is faster. So decomposition, typically, especially in fast decomposing leaves, it's an negative exponential. So meaning that in the beginning, decomposition is much faster and it slows down towards the end. This is because the fast decomposing compounds of the leaves disappear here fast, and then we only have the slow decomposing ones. For instance, the nerves of the leaves that are more resistant to decomposition. So when we put them, when they fall in the terrestrial ecosystem first, these are the fast decomposing compounds go away fast. And when they reach the water, they already have a small amount of these fast decomposing components and more of the slow decomposing, like the nerves of the leaves. So of course, decomposition will proceed slower because part of the decomposition of the material was already gone while they were in the terrestrial ecosystem.
So even they have harder, let's say, surrounding to survive and start the decomposition in the water, if I understand. Okay, but that's interesting. That's really nice.
I also thought so, but I did not convince anybody because nobody asked me anything.
Any questions from our audience? They are a little bit shy, maybe.
Maybe.
But I have the last one. I have a last one. So my question would be based on the results that you showed here in the beginning, and then the results about the aquatic terrestrial mix. Do you think that is there any good strategy? I'm a forest manager, So we are somehow encouraged to plan ahead the species composition of our forests. We base our decision on multiple factors, but we have some leeway. We can adjust it a little bit here and there. So, for example, is there a good strategy of planning species composition along streams and rivers? Should we follow some procedure, or should we leave it all to nature? What's your opinion?
Well, if you leave to nature, I think it already will happen what is necessary. Of course, I did not say here, but the decomposition process is very important because it fuels the aquatic communities. Of course, that's the main point. And also it contributes for the biogeochemical cycles and everything. But the aquatic communities depends on this leaf litter. The thing is, in our climates, yours and mine, we have a seasonal climate, and leaf fall, most of the broadleaf species, leaf fall occurs in autumn, meaning that in autumn they have a big amount of leaves that fall into the water, but during the rest of the year they will have very few food. Okay, so the most important thing is that we have a mixture of fast and slow decomposing leaves in the riparian area. If you have, so what happens in nature generally is the leaves fall more or less, not all at, depending on the species, not all at the same time, but they will fall in autumn. Okay, the fast ones, the fast decomposing ones, will be eaten faster and disappear, but if there are also slow decomposing ones, they will stay longer and they will feed the community during most of the year.
Okay, but from our experience or observations, we see that typically as a first succession, we get those, let's call them monocultures. I mean, if we have a stream, it would typically be alder coming in, and it's 100% really alder or poplar or whatever, right? But it's one species typically in the first stage.
But just one species. One species is not good. Alder, it's the preferred. I also carried out experiments with animals, offering them different types of leaves. And both animals and fungi, they prefer alder. Alder is the top food for aquatic ecosystems. The problem is that alder disappears really fast. So in an experiment in here, alder can disappear in 20 days, while poplar may take 40 days. And if we speak about quercus, it may take 180 days to disappear completely. So alder is really good, but if it's only alder, then the rest of the year they will have nothing to eat or very few.
And did you encounter any research on so-called alien species? Because, for example, in our ecosystems, we face, I think it's already a huge problem of Alnus glutinosa. Acer negundo, which is a very, it's kind of invasive. It takes over space along the rivers, along the streams. It grows fast. It's, the spread rate is very high. And we don't know, maybe in terms of what we are speaking right now, a little bit of it, it's not bad. But is there any research about?
I also made, but some other people are doing a lot of research on alien species, of course. But I also made some experiments, but using the species that are most common here, where I live.
So eucalyptus.
Also with eucalyptus, yes, but with the riparian species, acacia. Okay. And in the case of acacia, it's interesting because it's an alien species. It makes very thick stands, woody stands of things, and it completely replaces everything else. And acacia, it decomposes relatively fast because it's also a nitrogen fixer like alder, like Alnus glutinosa for the other one. But it also has other compounds that inhibit. So for one side, it has a lot of nitrogen, just like alder. But the good thing of alder is it's very rich, very high nitrogen, but it has very low amounts of secondary compounds. And so that's why it's so preferred. Some of the alien species, at least of what I know, they might be good also in the nitrogen levels like this one, but they have always some other secondary compounds that inhibit either the microorganisms or the invertebrates or both. Of course, invertebrates will eat if they don't have anything else, but it's not their preferred thing. But the biggest problem, I would say, is if we have a highly invasive species just like acacia and it replaces everything else, then you don't have a mixture. You only have acacia litter. And in the case of acacia, for instance, it interferes with a lot of things. For instance, the chemical properties of the water change because the leaching of the compounds alters many things inside the water, and including nitrogen. But high levels of nitrogen, dissolved nitrogen in water, are not good. So you see, it contributes to a lot of bad things. But mostly in the perspective of what happens to the aquatic communities, one species is not good because it does not give this continuum for the whole year.
So can we sum it up by saying that biodiversity matters here as well?
Yes, of course.
Okay. I see no questions from our
Nobody
attendees.
asked me anything.
So in case you would like to ask question to Manuela later, I think that you can contact her using, for example, email. The email should be somewhere in the invitation that you got for this meeting. But as for now, I think that we should slowly finish the discussion. And thank you, Manuela, for this really interesting topic. It was really nice to listen.
Thank you for the invitation.
It was a pleasure. And maybe I'll just show you one slide from my side. That
Okay, so I stop.
We already have a presenter for next month's seminar. So our seminar number 3 will be run by Marta Czcianowska. She's our researcher from Warsaw University of Life Sciences. She's, I think, on the other end of spectrum from ecology. She's in the applied sciences of wood logistics, but still in this area of forestry, agroecology, and then she now is working on the impact of wood logistics on the environment. And in next month, on the 20th of January, she's gonna show us how to design a wood yard to get the best performance. And I think it will be a totally different way of seeing things, but still, I hope that we can have a nice presentation, nice discussion afterwards. So I invite everybody to join us in January. And basically, that was my last information for all of you. So once again, it was a pleasure having you, Manuela. I hope that everybody learned something. I did for sure. And I hope to see you guys next month. And have a good day. Have a good time resting during Christmas, and see you in New Year's.
See you in January. Goodbye.
Yeah, bye-bye.